Okay, welcome to another video lecture. This time we're going to be focusing on the phases of meiosis, and throughout the lecture you're going to be noticing some similarities between meiosis and mitosis, and I will point out the differences that occur between the two. So let's look at the basics of meiosis, and you're going to notice in every corner of every slide, excuse me, we're going to be looking at PMAT, and hopefully you'll remember that that's prophase, metaphase, anaphase, and telophase. So let's look at what the cell goes through. I'm going to be referring to a term homologous pairs or homologous chromosomes. You can also use the term homologs, and that was from our previous lecture. So we're going to be referring to those terms primarily when we look at meiosis 1, and I'll explain what that means a little bit later. So we're going to be having these homologous pairs. You're going to receive one from the mom, which is maternal, and one from the dad, paternal. So this point is very important. In the past, it seemed to trip students up. It was a question on the exam, so do make a note of that in your lecture notes. So the chromosomes replicate during the S phase preceding, which means to come before, meiosis. So let's look back to the cell cycle. That's referring to the S phase. So the cell cycle, remember, has three main parts: interphase, which takes up about 3/4 of the cycle, and then specifically as G1S and G2. And then meiosis, but when we looked at cell cycle, that was for mitosis, but still the same on the cell cycle. Meiosis will go through PMAT1 and PMAT2, and then finally cytokinesis. So when we're talking about chromosomes replicate during the S phase preceding meiosis, we're talking about is coming before. So we're going to be focusing on what the chromosomes are doing before we start the process of meiosis. So at the beginning of meiosis, the cells are going to be diploid, which is 2N. And if you need to refer back to the previous lecture or your lecture notes in your spiral, then go ahead and do that. So it's going to be the total number of chromosomes. So let's start with the first phase, which is prophase. And so look at, I've got a Roman numeral here. And that's very important that you distinguish between prophase 1 and prophase 2. If you just put prophase, then you're not real sure communicating which part you're focusing on. So during this prophase 1, we're going to be looking at the homologous pairs. So they're going to start coming together, and this is the only time that you see what's called crossing over. And this is where we get our genetic variation. So remember, we're getting some of the information from our mom, some of the information from our dad, and when they cross over, that's where we get the variation, for example, between siblings. Okay, they'll have some characteristics, but there may be some slight characteristics that are different between brother and sister or sister and sister. So here's some images, and I always like to put the actual image of what's going on in the cell and a drawing. So here we see the homologous pairs. So just like a pair of shoes, you want to match up similar chromosomes. So we have the same length. So here are some short chromosomes, you notice they're the same. So that's a pair, homologous pair, and these are a little bit longer. And I'll have a little bit larger slide, but here they are going through the process of crossing over. So during crossing over, and there's a good illustration in your book, your online textbook, you may want to take a look at that also. But just to make it simple, we're going to go ahead and label the genes. So on this particular sister chromatid, we have ABC lowercase, and on this chromatid we have ABC uppercase. And when they're going to go through replication, okay, so here they've replicated. And here they've replicated. Now I've got the complete chromosomes, and now I have a homologous pair. So during prophase 1, we have crossing over. And basically, one of the ends, and it could happen up here, if this was switched and the lighter color green was on the other side, it doesn't matter. It depends on how they pair up. We have the tips are going to cross or lay over each other. It kind of looks like they're holding hands, so to speak. And when they do that, when they recombine for recombination, the piece is going to break off in exchange. So if I come down here to my gametes, okay, so which means that each cell, so obviously we're making sperm and egg cells, and that's where you find meiosis, and I should have indicated that at the beginning. So these are 4 egg cells or 4 sperm cells. So we can see here that this particular chromosome, which will condense and it will combine with the egg or sperm depending on what this is. But here I have the combination of A, B, lowercase, and a capital C, and then capital A, capital B, and lowercase c. So when they combine, and we're going to get to genetics in the next chapter, then we'll look at which is going to be the dominant, recessive, codominant, etc. So crossing over only occurs prophase 1, and that will definitely be a quiz on the exam. Next, following PMAT, I will have metaphase 1. And just like before, it's still going to go through the same process of lining up at the equator of the cell. Okay, but this time it's the homologous pairs that are going to attach to the spindle fiber. Very important, okay? So that's why you have to distinguish if it's 1 or 2. Then I have anaphase 1. And just as mitosis, this time instead of splitting the chromosome into chromatids, I am separating the chromosomes. The homologous pairs are going to separate. So 1/2 of that pair is going to go toward the centriole, and the other half is going toward the centriole. And if you can see here, we have 2 different colors. That's as a result of crossing over. Okay? Then we have telophase. And at this time, we're going to go through and start, it'll finally factor in cytokinesis. And this is where we're going to divide the cells. So its reproductive cells are dividing. And that's how we eventually will end up with 4 cells, 4 gametes. Oops, sorry. So at the end of the first phase of meiosis, I will have divided the homologous chromosomes, and the diploid cell will now become 2 haploid cells. So I'll end up with 2 haploid. Okay, then it starts the phases over, but this time I indicate that it's a 2 with the Roman numeral 2. This is going to be exactly like mitosis. So exactly like mitosis. So now I'm going to go ahead and rearrange, kind of get things back in order. And sometimes you may find books where the authors or the scientists will actually leave this out because it is such a short cycle. One key thing is that there's no crossing over in prophase 2. That is only prophase 1. Metaphase 2, we are going to line the chromosomes up down the middle of the equator, and the centromere is attaching to the spindle fibers again, just like mitosis. Anaphase 2, we are separating the chromosomes, and now they're called chromatids, and they are being pulled toward the centrioles. Telophase 2, this completes the cycle after we finish up cytokinesis. So we've divided the sister chromatids. We now have 4 haploid cells, and each will contain half the number of the original chromosomes when we started with prophase 1. So we have 23 in each of these cells, so it could be sperm, it could be eggs. Male and female both go through the same process. We'll look in just a second on how the cytoplasm, when it goes through cytokinesis. The females go through a little bit different. Okay, so for the egg formation, now the sperm will all get equal number of cytoplasm, which means that all of the cells are viable. But in females for meiosis, it starts off, and we'll talk more about this a little bit later. You may have noticed the term oocyte. But all I want to point out here during this lecture is how the cytoplasm is redirecting to one particular haploid cell after the completion of prophase 2. And the other ones do not get cytoplasm, so they cannot be fertilized because you need to have the cytoplasm for all of the organelles to be functioning. So it's dried up and it's non-existent. So all of the cytoplasm will go to one, and it doesn't necessarily have to be this particular haploid cell. It could be any of them. However, if this is the polar body, when it replicates, it still goes through replication, then it's not going to have any cytoplasm. But on this, it could have been this particular cell, or it could have been this one. And then that will develop into the egg. So that's all that I'm going to cover for this particular lecture. Hopefully that helps. And please, I encourage you that during class, the reason that I do this on the video lectures at home is so that if you have questions in class, we can sit down one-on-one and actually go over this if you're confused at any point. So please feel free to come and ask me any questions. Okay, so that wraps up this lecture. Thank you.